lecture. I guess integration metabolism, there's no board questions. Is that the only one?
Yeah.
Okay, so the other ones, you know, when I look through the Scantron results, you know, it's like 95%, 80, 86, 90, 100. So, yeah, so those are, so you'll have those. So that comes down roughly to 4 questions per lecture. And everyone should do better because, you know, it's not this horrible exam to pile up, so people have a little bit more time, not quite as burned out. And I think the average, I'm going to say it again, Jonathan, I think the average is going to be really high. So maybe I'll be proved wrong again.
All burned out.
Yeah. So if you didn't do well on the first 2, or especially the second one, don't come into this with a defeatist attitude. Because I think it's going to be fine. So, okay, so then on to the content. I guess we can just march through it lecture by lecture. So the first one, catabolism of amino acids. So we're just talking, you know, biosynthesis of amino acids is what was the word, tedious? Yeah, it's tedious. And I agree. Catabolism is, I think it's a lot more interesting, makes a lot more sense. So there's kind of, I guess, 3 big notions. The first part is, you know, this role of glutamate dehydrogenase. How do we get the ammonium or the amine group out, right? So that's glutamate dehydrogenase. So that's getting it out. The other part is how do we, you know, we're not going to just break down glutamate. So how do we shuttle in the other amino groups through transamination? And then sort of the third key part is the urea cycle. You know, once we get it out, what do we do with it? So we can, I think, just talk about those 3 main concepts. So glutamate dehydrogenase, we see it again in the biosynthesis lecture, right? Because it's completely reversible. And I apologize to those that come to the office and we talked about this at length. But bear in mind, there's only one amino acid dehydrogenase, and that's glutamate. So it's going to be our entry and our exit point for ammonium or the amino group. And then in between, it's going to be transaminases. And then what do we know about transaminases? Well, there's a alpha-keto acid counterpart. What do we know about this enzyme? What cofactor does it use? PLP. So it's holding, so the alpha amino acid comes in, the alpha amino group comes off, the PLP covalently holds it, an alpha keto goes off, another alpha keto comes in. Could be that the previous alpha keto came in, nothing happened, but if a different one comes in, then that alpha amino group is then transferred back to that alpha keto, and voila, you've got a new alpha amino acid. So you can shuttle around, and then if you're trying to get rid of amino groups, then the one that you're making is glutamate. And then it can be oxidatively deaminated to free the ammonium for the urea cycle.
So glutamate dehydrogenase only takes it from glutamate to alpha-ketoglutamate, right?
Right, and so, you know, so the point is that glutamate dehydrogenase is, you know, it's just glutamate and alpha-ketoglutarate. It's not a transamination. And as well, it involves oxidation and reduction because, say, if you're bringing it in to alpha-ketoglutarate, you have to also bring in the hydride ion of the alpha at the alpha carbon, right? You're bringing in the amine and you're bringing in the hydride ion so that you make an alpha amino acid. And then when you're going out, you're removing the amine and you're removing the hydride ion.
Glutamate dehydrogenase.
It's freely reversible. So one enzyme, so all cells have one enzyme to bring ammonia in and to take it out.
So it does, so in essence it does oxidative deamination and reductive amination.
Right. And then the other weird aspect is it can use NAD or plus or NADP plus, so that it doesn't discriminate. So that's a big concept. There's only one amino acid dehydrogenase, and that's all you need, because once you have that, then you can just exchange to these transaminases covalently. Okay, so what else? So in the urea cycle, so we know that starts in a compartment. So the ammonia is coming off of the glutamate through the dehydrogenase, and it's in the mitochondrial matrix. And so then the first step, we see this again where?
Purine synthesis.
Purine synthesis, right. So, and it's actually catalyzed by an isozyme. So these are isozymes, very structurally similar, mechanistically similar. It's the exact same chemistry. So you're just simply condensing it then through how many activation steps? Two, because you have to first activate the CO2 to bring in the amine as a nucleophile, and then that's no good to you unless you activate that again, so it can then condense with an alpha amino acid as part of the cycle. I was just looking at that slide. I thought it was interesting because when you look at it, the first step, you've almost got urea, and you took CO2 and you added an amine. Why can't you just take that and add another amine to it and you're done, right? So you're almost done, but no, you've got to go through this big, long cycle. So all I can figure is that this enzyme, carbonyl phosphate synthase, that's used for these dual biosynthetic purposes. You're making urea for catabolism, but at any rate, you can do this, but you can't. There's no enzyme to put this other group on, so you have to go through this long, convoluted process. I don't know. That's the way I rationalize it. But so what's our bird's eye view of the urea cycle? There's a lot of chemistry going on in there. It all makes sense, but it's a lot. So we have this activated carbonyl phosphate. It's essentially an activated amide. It's just an amide group with a leaving group that we can then condense with something. So what is the next step? It condenses with what? Ornithine, but what's ornithine? It's just an alpha amino acid with a funny name. So it condenses, okay, and then our next step is we're going to bring in that other amino group, and it's going to come from where? Aspartate. So it condenses, and now we got something with a really funny sounding name, but what is it generally? It's another alpha amino acid, but then when that breaks down, what do we generate? Fumarate, and that's good because it links us to the TCA cycle, and eventually oxaloacetate. And then that produces an amino acid that, that could be biosynthesis of that one, and then we harvest the amino group, right? To eventually then transfer to to give us our final product. So that's my view of the urea cycle. I guess I could recall the names. What's the first one is ornithine, and then citrulline, and arginosuccinate, and arginine. Did I finish that off, or I cut it short?
It's pretty good.
It's a, okay, but I mean in terms of just the cycle, did I? Yeah, that's just, yeah, so, okay, so there wasn't anything, I guess there's not, is there anything after that in the lecture, after urea cycle? I didn't remember.
Just the fates of
the fates. Yeah, okay, and then we looked at that later in the integration of metabolism. So we can maybe talk about that at that point. Okay, then as well, then what do we need to know about metabolizing amino acids? What's just the general? What's the gist of it? What can happen to amino acids?
3 different things.
Well, there's 2 general fates: ketogenic or glucogenic. Right. So, and then it turns out, are they all? Is it glucogenic? Are they all glucogenic? So there's 2, right? So the other way to look at it, are they all ketogenic? Yeah. So they're all ketogenic. There's just 2 that are at a place where you can't go into gluconeogenesis, and then it just lysine and leucine. Just happen to memorize those. But
Okay, all are ketogenic? Okay,
right.
Sorry.
Yeah.
Don't do that.
Yeah. So
Wait, you said not all of them are ketogenic.
no, they're all ketogenic. So there's 2 that you can't use to get to glucose.
Sammy, now I can't figure
Yeah.
it out.
So that, for me, that's good enough. And you know, that's just because those, for some reason, you can only get to acetyl